Hi everyone, and welcome to the last of our lectures. This one focuses on diagnostic microbiology and antibiotics. Now comes the fun part, which is diagnosing infections, and some of these ways I know they're going to sound kind of complicated and not so fun if you don't have a strong background in things like DNA and RNA, but what we'll do is we will basically give a brief summary of each, okay? So we're not going to go crazy in terms of details and specifics. That's more for advanced micro, okay? Now, what I want you to be aware of are the three main ways of diagnosing infections and be able to briefly explain each one, okay? How exactly can that diagnostic technique be used to show whether or not a patient has a particular infection? Okay, so the three main ways we have listed here are growth analyses or biochemical testing, which is the first one. And you'll notice this first one, growth analyses and biochemical testing, is actually what we do in microbiology class lab, okay, when you work with things like selective or differential media. So basically, growth analyses and biochemical testing is looking to see how different organisms grow on different types of media to help narrow down or identify which infection is present. Serology is going to be the antigen antibody test that we'll look at in a minute. Okay, so whenever you hear serology, think antibody and antigens. And then the last one is my personal favorite, nucleic acid-based detection. And whenever you hear nucleic acids, think DNA. Okay, so we're going to go one by one through these. And again, as we go through these, make sure you're comfortable explaining some of the ways in which these can be used to diagnose a patient. Okay, so the first diagnostic category, as we said, is growth-based diagnostics. And this one is the easiest and cheapest one to do. As you saw, you're even able to do it in a college lab classroom setting rather quickly. In the growth-based diagnostics, you're basically dealing with the idea that microbes will have different characteristics when they're on different types of media based on their metabolic processes. So you have the idea of selective and differential media. So with selective media, again, that's the idea that on this media, the microbe will either grow or not grow. So certain strains will be inhibited, or certain species will be inhibited. And then you have the idea of differential, where depending on which organism is present, you'll have all different colors. So for instance, the idea of the MSA plate, the differential aspect is how you get the different colors depending on whether or not Staph aureus is present. So you see Staph aureus turns the media yellow. Okay. The other type of growth-based diagnostics that you see on this slide here is the enteropleury tube. And again, remember the enteropleury tubes are only able to diagnose gram-negative strains. Okay? So depending on which organism you think is infecting the patient would determine which types of media you would use. There's also serology-based diagnostics, which will also be used in microbiology classroom lab. The example that we use in lab is ELISA, but there are various types of serology-based diagnostics. Now, whenever you hear serology, I always want you to think right away, antigens and antibodies. So remember that antigens are the proteins that are like name tags on the surface of pathogens, whereas antibodies are what the human host cell makes. Okay, the human host body will make antibodies specific to the pathogen's antigens. Okay, so now when we talk about serology-based diagnostics, the way you look at it is if a patient is infected with a pathogen, then that pathogen is in their body. So they now have that pathogen's antigens present in their body, and their body has made the specific antibodies to fight that pathogen. So you are basically looking to see, does their body samples, so their blood samples, their patient serums, their cultures, have pathogen antigens or even the antibodies against them. Either one will show you that the specific pathogen is present. The three examples that I want you to know for serology-based diagnostics are the slide agglutination test, Western blot, and ELISA, which ELISA is the one that you will be performing in this course, and it's called the enzyme-linked immunosorbent assay. Now, the easiest one to understand or to explain, if you're ever asked to explain one of these tests, is the slide agglutination test. So what you see here is the slide agglutination test, and in this test, basically what happens is you place a certain amount of antigen, pathogen antigen on this slide. So for instance, let's say you think that your patient has been infected with E. coli. You would put E. coli proteins on this slide. You would then apply the patient's serum on top of that and mix it together. What you then look for is, do they react together? Do they clump up? Okay, because what that would tell you is, if you have on this slide the bacterial or viral proteins, right, the antigens, and you apply the patient's serum, well, if that patient is infected with that particular pathogen, then their body has made the matching antibodies. And as you know, antibodies and antigens like to bind together. And that would lead to the clumping, which you see in this positive test result. Whereas if you have the antigen on the slide, and then you put the patient's serum on top of it, and that patient is not infected with that particular bacteria, then there's no antibodies present. There's nothing to bind and clump to that antigen. So it just stays a nice even smear. Okay? So with the slide agglutination test, again, if you had to explain it to someone, all you're doing is putting known bacteria antigens on the slide and then applying the patient's serum. If the patient's infected, they will have the matching antibodies and you see clumping. If they're not infected, there's no antibodies, so there's nothing to bind or clump together. And by the way, agglutinate or agglutination is a fancy way of saying clumping. So here you have the recap notes in case you didn't get to write down what I was saying with regard to the agglutination test. So please pause the recording and make sure to write down these notes to be able to explain what exactly is occurring in this serology-based diagnosis. Another example of serology is Western blot, and you see this a lot in research. So with Western blotting, it's a similar concept in that you're looking to see, does your patient have the antigens or antibodies that match up with this particular bacterial infection that you suspect. So in the case of Western blot, what you would do is you'd put all of your proteins from the patient onto a gel, okay? You would then have that gel transferred onto a thin membrane, a membrane that basically has pores to pick up all of the proteins from that gel. You would then block that membrane so that there's no what we call non-specific binding, and you'd add a primary antibody against the suspected pathogen's antigen. Okay, because here's the logic behind it. If your patient is infected with that particular bacteria, right, then in their protein sample, they'll also have that bacteria's proteins, right, because they have that bacteria floating around in their body. So now, in their protein sample, you would have the bacterial antigen, and you apply a primary antibody that matches that antigen. So if they're infected, your primary antibody will stick to the membrane as well. If they're not infected, the primary antibody won't find anything to stick to and will get washed away. So now what you then add is a secondary antibody. And secondary antibodies are special in that they'll bind any antibody, but they also on their other side will have some sort of detection signal. So as you see in this one, the detection signal can either be color change or fluorescence. Okay? And so that will allow you to see whether or not your antibodies bound. Again, if they bound to this membrane, then that means your patient's infected and their body has the bacterial or viral protein antigen present. If they did not bind, then there's no matching antigens for that suspected infection. Okay. The last one for serology is ELISA, and that's the one that you'll perform in lab. This is a similar concept to Western blot, except now it's performed in wells or even in flat strips. Okay. Now with this, what you do is you coat the bottom of each well with an antibody, so that you see here, an antibody matching the suspected antigen. You then add the patient's serum, and if they're infected, then they'll have the antigen that sticks to that antibody. You'll then apply another antibody that has detection on it, for instance, color changing detection. And if you see that binding and you get the color change, then you know that that patient is infected. Now, what I want you to remember for ELISA is the fact that pregnancy tests are actually an example of ELISA. Okay, so we'll see that in the next slide, what we mean by that. So over here, you see a pregnancy test to show you how the pregnancy test matches the concept of ELISA. And I always basically say that the pregnancy test, ELISA, is testing for the biggest parasitic infection that you can get that you're stuck with for life. Now with the pregnancy test, what you'll notice is it's testing for the presence in the patient sample of what's called HCG, which is a hormone that's only produced in high levels if someone's pregnant. So now what you do is you have this strip where one portion of it will have antibodies that are very specific to that HCG. Okay? They get pulled through the strip as the urine sample or whichever fluid sample is going through, and they bind to this area where the antibodies are specific to HCG. You then also have antibodies at the very end of the test that are general, that will bind any antibodies, including the ones that get pushed across the strip. This is just to show you a control of what the positive results should look like. Okay, so one more time, just to make sure this is all clear, if you look at it from this perspective, rather than on this diagram, you can see that the first portion of this ELISA strip will have a whole bunch of empty antibodies specific to whatever you're testing for. So in this figure, it's testing for this particular respiratory virus. In this area, the antibodies that would be present are specific to HCG. Okay? You then apply the patient sample, which will pull through capillary action these antibodies across the strip. If the patient is infected, then their sample will have the antigen or proteins that match the antibodies, get stuck to it, and they will then bind to the test strip area, where you'll see a color change. More of them will also pass through to the end portion, which is the general control antibodies that will bind the original antibodies no matter what, just so that you can see what a positive result would look like. If the person is not infected or not pregnant, then what happens is the antibodies from the beginning do not get any protein bound to them. No antigen binds them. So when they float through here through capillary action, they're not going to bind to the test area, okay, because there's no hormone, no antigen to bind here. They'll keep floating through, and then they'll bind to the control area because this control area is meant to stick to any antibody. So you get to see a positive result as a control to show what it looks like. And then you would compare and say, oh, my test area does not look like the control, so I'm not pregnant. Okay? If this isn't completely clear right now, don't worry. In lab, we'll go through a little more details, and you'll get to really visualize exactly what's happening. Also, feel free to contact me if you're having difficulty understanding any of the diagnostic tests that we talked about. The last category is usually the trickiest, and I'm not going to go into the crazy details of it because you don't need that for this course. Again, we go into more detail in advanced micro. What I need you to understand is the idea that nucleic acids can be used for diagnostics. They will be the most accurate, but also one of the more expensive ways to diagnose the infection. There are a few different techniques that can be used that rely on DNA. The first one is PCR. If you take any genetics or molecular-based courses, you always get to run PCR, which stands for polymerase chain reaction. Now, what PCR is, in the case of diagnostics, is you'll have your clinical sample, okay? So, for instance, a patient's sample. Now, if your patient is infected with a particular bacteria or virus, you'll have the pathogen's DNA in addition to the patient's DNA in that sample. Okay? So what you then do is denature that DNA sample into single strands. And what do you know about single strands of DNA or RNA? Singles are clingy and want to find their match, right? So if you denature their DNA sample so that it's all single-stranded, and then you apply these little probes, meaning these small single-stranded pieces of pathogen DNA that have little fluorescent detectors or some kind of detection on them. What then happens is, if the patient's infected, these probes will find their match in the sample, and they'll bind to it. You can then amplify them and get a detection. So for instance, you run them on a DNA gel and you'll see the UV bands light up. Okay. What you mainly need to get from this is the fact that PCR, polymerase chain reaction, is an example of nucleic acid-based diagnostics. And also I want you to remember the fact that all of these nucleic acid diagnostic techniques are relying on the fact that single strands of DNA will try to bind to their complement. Okay, so that lets you probe for specific sequences. Another type of nucleic acid-based diagnostics that you'll hear about are things like hybridization or microarrays. Okay, so again, you don't have to know or understand all of the little details of this technique. Simply understand that what you do is you have the patient sample with the DNA release. You turn that DNA sample into single strands, and then you probe with single-stranded fragments of a sequence that matches the suspected pathogen's DNA. And those small sequences will have some sort of detection. So in this case, the small sequences, these little probes, have fluorescence. So if your patient has the particular pathogen's DNA, so in this case it's Salmonella DNA, this single-stranded little fluorescent probe will bind to your patient's sample of DNA because they're infected with Salmonella. You'll then see the fluorescence through that binding. If they are not infected, then the fragment will never find a match to bind to, and you won't see any fluorescence because they're usually made in a way that they only fluoresce when they're bound to their partner, and otherwise they get washed away throughout the technique. Okay, one more example of this, microarrays. So one more example of nucleic acid-based diagnostics are microarrays. It's the same concept that I was just talking about. Just now it's on a specialized computer-looking chip, okay, kind of like you can see here. And again, it's that same idea where you will have these small fluorescent probes looking to see if the pathogen DNA is in the sample of patient's DNA. And if the patient is infected, then the pathogen DNA will be present, it will stick together with its match, and you will see fluorescence. Okay, so that's showing the fluorescence that you will see. Okay, if the pathogen is not present, if that suspected pathogen is not present, then the fluorescent probes get washed away during the process. Okay. So again, just to recap for what you need to know about nucleic acid as diagnostics, really focus on the examples. So know that PCR and microarray are examples of nucleic acids based diagnostics, and also understand that all of this is working because of the concept that single-stranded pieces of DNA like to find their match and stick together. Okay, so basically if a patient's infected, then their body will have the pathogen and the pathogen's DNA in it. And so if we denature the patient's DNA samples into single strands, we can see if pathogen DNA single strands will stick to that. And then we always have some form of detection such as fluorescence. Okay, so again, you can pause this slide and write down these notes just in case you ever have to explain what exactly nucleic acid-based diagnostics means. Now, the big term for today's lesson and throughout the course of microbiology is selective toxicity. It is a fundamental criteria for picking any kind of medical antibiotic or drug that you are going to use. I want you to circle, star, highlight 1,000,000 times the term selective toxicity, and make sure you know its definition. The definition is selective toxicity means you should destroy the pathogen with little or no harm to the host. Okay, so destroy the pathogen with little or no harm to the host. Okay, so anytime we talk about selective toxicity, things that are the most selectively toxic will be the best at hurting the pathogen without hurting the human or the host, whereas the least selectively toxic targets will end up hurting both the pathogen and the host. So you don't want anything that's least selectively toxic. You want your drugs or antibiotics to be very selective for only hurting the bacteria, selecting the bacteria and not the host. When we talk about that, there is one structure you have to remember, and that is the cell wall. The cell wall, out of all of the groups listed on this slide, or all the groups you can think of in terms of targeting to kill or hurt bacteria, the cell wall is the most selectively toxic. Okay, the most selectively toxic target is the cell wall, and that's because, as we mentioned previously, bacterial cells have a cell wall, whereas human cells do not. So you can pump a chemical or drug into the human body that destroys cell walls, and since their own cells do not have this, it's not going to hurt their own cells. The least selectively toxic target is the plasma membrane. Okay, so that is the worst one to target because our plasma membrane is identical to bacteria's plasma membrane. So when you target the plasma membrane, you're also going to hurt the human. Okay, so make sure you write most selectively toxic next to the cell wall and least selectively toxic, bad to target, next to the plasma membrane. Some antibiotics and drugs will also target some other things such as protein synthesis, nucleic acids, or the metabolism of the bacteria. But again, the best one to target is always the cell wall if possible. This slide is just to give you some examples of various common antimicrobials or antibiotics that you'll encounter when you're in the medical field, or again, if you yourself ever end up in any kind of situation with infections. And it shows for each of those common agents what is targeted. And you'll notice that these three, including the very first famous penicillins, they target the cell wall. Whereas these guys here, such as tetracyclines, instead they target the ribosomes, which is protein synthesis. Rifamycins and quinolones. I want you to put a star next to rifamycins and quinolones and make note that those target nucleic acids, such as RNA and DNA. Now, when targeting nucleic acids, again, these structures and their enzymes are so similar to humans that these guys are also going to cause some problems for the patient as well. So whenever you're dealing with any rifamycins or quinolones, you're going to see a huge list of side effects. Okay, so they are still used because they're possible to target, but they're usually a later resource if initial antibiotics did not work, or if your patient or you are suffering from a really intense infection. Okay? The other one listed here is sulfonamides, and we'll talk about folate or folic acid metabolism in a minute. Now, as I already mentioned, the cell wall is your number one best target. It is the most easily selectively toxic target because, like we said, bacteria have it, but we do not. So you can destroy the cell walls all you want without destroying the human cells because the human cells do not have the cell walls. The two examples to remember, and please do remember that they are examples of cell wall targeting, are penicillin and cephalosporins. Okay, penicillin and cephalosporins, what they do is they block the enzymes that build peptidoglycan, specifically the ones that will make that matrix, that cross-linking of sugars that we mentioned previously, the NAG-NAM sugars. Okay? The way they do this is, if you look at their chemical structure, they have what's called a beta-lactam ring. Okay, beta-lactam ring. I want you to circle the word beta-lactam ring because that will be important when we talk about resistance. Okay, so basically what they do is they use that ring to then bind to the enzymes that would build peptidoglycan so that those enzymes can't work anymore. No peptidoglycan, no cell wall. Now, very important question I wrote over here. For which bacterial infection won't it work to target the cell wall? Well, which bacteria do you know of that does not have a cell wall to target? That would be mycoplasma, if you remember mycoplasma. And so the infection that this wouldn't work for is walking pneumonia, which we said is caused by mycoplasma. Okay, so make sure to write both down as the answer. So it will not work for walking pneumonia caused by mycoplasma. Now, the next structure that you might want to target would be plasma membrane, but remember we said, do not target this. The plasma membrane is the least selectively toxic target. Okay, so this one is not so great to choose. Why? Because our plasma membrane is the same as bacteria. So if you put something in the human body that is destroying bacteria plasma membranes, it will also destroy the human plasma membrane. So it'll destroy your own cells, which you do not want to do. The next option would be to target protein synthesis. Now you may be saying to yourself, well, I'm pretty sure my cells have to produce proteins too, and they both have to use ribosomes, right? Well, lucky for us, the bacterial ribosomes are slightly different in structure from ours. So if you look at this little figure I made, it's basically fancy scientific ways of showing that the prokaryote or bacterial ribosome is a slightly different size and shape and made of slightly different components than the eukaryotic one. Okay, so you don't have to worry about the exact numbers or the components. Just realize that bacterial ribosomes are slightly different from eukaryotic ribosomes, which allows us to target them if we want. But again, it won't be as good as targeting the cell wall because, the cell wall we don't have at all. The next option is to target nucleic acids. And like I said a few slides ago, I want you to put stars next to quinolones and rifamycin. And make sure you know anytime you hear those words or you see them in writing, right away think to yourself, these target nucleic acids. So either DNA or RNA. Now, instead of targeting the exact structures of DNA or RNA, since ours are identical, made up of the same A's and G's and C's, instead, these guys target enzymes involved in the process. But now, since I've already said that, we both have DNA, RNA, and similar kinds of structures and all, you will see a lot of side effects with these. So targeting nucleic acids is a problem because nucleic acids we have similarities with. So humans and bacteria have similarities. Now the reason why I want you to star and remember quinolones and rifamycins targeting DNA synthesis, RNA synthesis, enzymes such as topoisomerases or bacterial RNA polymerase is because the side effects will be so problematic. So the example I always use is about a year or two ago, my grandmother had a really bad infection and the first round of antibiotics didn't work. So my grandfather called me after he had picked up the new antibiotics for her, and he was very concerned and worried because he said to me, wow, there's a really long list of side effects with this booklet, with this prescription. He said it's like having a book attached to it with so many side effects. And I asked him, I said right away, does it say quinolones or rifamycins anywhere? And he goes, yeah, it says rifamycins on it. I knew right away, just from hearing the fact that there's a long list of side effects. I knew exactly what they were giving and why. So the fact that the first round of antibiotics didn't work, they then had to switch to a stronger, but possibly more problematic antibiotic, which would be the rifamycins. But again, keep in mind, both of these, quinolones, rifamycins, are targeting nucleic acids, so they will have a ton of side effects. The last thing you might want to target is metabolism. And when we think about metabolism, we say all of the chemical reactions in a body or cell. So when we say targeting metabolism, you're using a drug or antibiotic to destroy one of the pathways, the chemical reactions within a bacteria. So something that it would try to build, you are blocking it from building or producing. So the one that we tend to target is the folic acid pathway. So circle the term folic acid. Folic acid, I'm sure everyone here has probably heard of because you hear it with humans as well. So the folic acid pathway is necessary in order to build or make nucleic acids. So folic acid is needed to make nucleic acids. So it's very important for bacterial cells and also our cells. Now, I just said that we need folic acid. So how is it that we can target the folic acid pathway and block production, but not hurt our patient? Well, where else can that patient get folic acid? Does the patient have to eat? Does the patient have to build folic acid? No, the patient can eat it, right? We can get it from our diet. We can get it from even pills. Think of if someone's pregnant, what do? They take prenatal vitamins that have a ton of folic acid in them. So yes, you can block the synthesis of folic acid and have your patient be okay, because the patient can still get folic acid from taking it in, eating it, or vitamins, okay? Whereas the bacteria, you're not going to see them eating a nice leafy salad and getting folic acid, okay? So when you think of our diet getting folic acid, some of the big places you get it are leafy veggies, citrus, beans, and rice. But like I said, also a lot of supplements like prenatal vitamins as well. Okay, so whenever you hear targeting metabolism, think folic acid pathway, and make sure you know why that's okay to target, since we need folic acid as well. Now, so far we've been focusing on antibiotics and trying to destroy bacteria. But if you think about it, not every infection is going to be bacteria. There are plenty of other infections as well, such as viral infections. And as you know, you can't give a patient an antibiotic for a viral infection. Now, viral infections are much more difficult to treat than bacterial infections. In fact, when we talk about any of the infections, I want you to know that viruses are going to be the most difficult to treat. We'll kind of put them in a certain order in a little bit, but for now, think viruses, most difficult infections to treat, okay? The reason that is that they are intracellular parasites. So if you look at that word and ask yourself what it means, when you hear something is intra, that means it's within. So this is saying intracellular, so it's within your cells. That means that these parasites go inside your own cells. They integrate a lot of times with your own DNA. And so trying to target them with a drug is going to be quite harmful to the patient as well, because of how integrated they've now become, how embedded within your own cells they've become. Okay, so most drugs are going to be toxic to the host, but you still have to try because of the damage that the virus is doing. So you hope that the drugs that you give will be less toxic than the virus itself, okay? But it will, any of the antivirals will have quite a few side effects and be rather dangerous for the host, okay? Now, with viruses, there are a few things that we target with antivirals, okay? If you think of them, when you think of viruses, they're basically just a protein shell around a nucleic acid, okay? They're not as robust or filled with items as bacterial cells or our cells. A virus is just two things. It's the protein shell with a nucleic acid inside, either DNA or RNA. So when we go to target them, there's a few things that they tend to target. One is the assembly and release of these viruses. Okay, so that means any of the pathways that the viruses use to build that protein shell and put together new viruses. Notice when we talk about viruses, we'll always say assembly because they do not reproduce, they don't have sex, they don't divide, they assemble. Basically what they do is they make more nucleic acid, more protein shells, and they put them together. So what we do is we try to block those pathways. Another thing we try to block are the viral DNA polymerases that are responsible for replicating their genome. Because like I said, if they're just a genome, a nucleic acid, and a protein shell, then you want to stop them from being able to make more of themselves. The last thing that we tend to try and target is viral uncoating or entry pathways. And again, this will make more sense when you really get into more detail of viruses, but basically this is trying to target things like receptors on your own cells that they may use to try and enter your cells. But again, no matter what you're targeting when it comes to antivirals, they're gonna tend to be kind of nasty for your patient and have a lot of side effects. Because again, viruses are the most difficult to treat as they are intracellular parasites. Now, in addition to viral infections, sometimes you or your patient will have fungal infections. Okay? Now, the good thing about fungi is that they do have a cell wall. And anytime you hear cell walls, what do you think of? Very selectively toxic. So now when it comes to targeting the cell wall, antifungals, they're going to be easier than viruses, right? Because cell walls are very selectively toxic. But fungi are more difficult to treat than bacteria because their cell wall is actually stronger than bacterial cell walls because they have what's called sterols, like ergosterol, within their cell wall, which is basically a more robust carbohydrate than you would find in the bacterial cell walls. Okay, so like I said, when you start to think about putting in order which infections are the easiest to treat, which are the hardest to treat in terms of targeting with drugs, the easiest will be bacteria, then fungi, because they also have a cell wall, but it's a thicker, tougher one, and then the most difficult will be the viruses. Okay. Now with fungi, sometimes they also try to target nucleic acid synthesis, but again, why do they not really want to do that often? Well, anything to do with nucleic acids, kind of know that's hitting a bit close to home, meaning the human, your patient, you, whoever's getting treated, also has nucleic acids. So it's riskier, going to have a little more side effects. Okay, so when you can target a cell wall, go for it. The last category of drugs to mention are the antiparasitics. Now, the antiparasitics are similar to the idea of viruses in that it's kind of difficult to find truly effective therapies for these guys because, again, they tend to be intracellular parasites. Now, they are a little easier than viruses to target because of some differences that they have from us and the fact that they do not integrate into our DNA. So viruses tend to integrate into our DNA and really embed within the cells, whereas parasites do not tend to become one with our DNA. Now, since they are difficult and they're intracellular, they will have a lot of really severe side effects like antivirals will. But there are some options for targeting when it comes to parasites. We tend to target asexual reproduction and folic acid metabolism. So asexual reproduction is very selectively toxic because think about it. Do humans reproduce asexually? No, they reproduce sexually. So targeting asexual reproduction shouldn't hurt your host as much. The other thing to target, like we've mentioned before, is the folic acid metabolism. Now, anytime you hear folic acid, remember there are a few things I want you to remember. First off, folic acid is used for making nucleic acids. It is needed by bacteria and humans, but the difference between us is humans can obtain folic acid from their diet. So it's okay for you to try and target the pathway that produces it, okay? Because we can still get it anyway. So now looking at this slide, I just want to kind of fill in the last piece of what we've been organizing so far. When we put infections in order in terms of difficulty to treat or find therapies for, the hardest to treat will be viral infections. The second most difficult will be antiparasitics. Next is antifungals. And then the easiest is bacterial. Okay, so the order is viral, parasitic, fungal, bacteria, from hardest to treat to easiest to target. Okay, so now that you have become comfortable with regard to antibiotics and drugs targeting infections, now we get to the big topic, the antibiotic resistance, which you hear all over the news, and if you're in the hospitals, you hear it within there as well, because it's becoming a true worldwide crisis. Now, the big fact that I want you to remember when it comes to antibiotic resistance, the thing that is causing this resistance problem is killing off normal flora. Okay, that's going to be the constant message that you get throughout all of these slides. Okay, that killing off normal flora is allowing the resistant bacteria to then flourish, take over, and become a problem. Okay? As you know, normal flora are supposed to be your good, healthy bacteria, keeping the bad guys from taking over. So if you kill them with antibiotics, then that's going to be a problem. So now we're going to go into more information about antibiotic resistance. Okay, so now as we continue to think about that concept of antibiotic resistance, there are a few questions that I want you to think about. The first one is something that we already touched upon, which is why would bacteria naturally have resistance to antibiotics? And the reason that they naturally have to have resistance is because they naturally produce antibiotics. So if they didn't have resistance, then they wouldn't be able to survive the antibiotics that they produce that they want to use to kill other bacteria who are their competitors. Now the next question is very important because this is the big deal that you constantly see on the news and the controversy in terms of antibiotic use. So why exactly does increased exposure to antibiotics increase antibiotic resistance? Now, the term I want you to remember here is evolutionary pressure. If you are increasing the exposure to antibiotics, you're increasing the evolutionary pressure towards antibiotic resistance. So what that means is antibiotic resistance, for a bacteria to have that, is only an advantage if the antibiotic is present, right? If the antibiotic is not present, then there's nothing that bacteria would ever notice. So what this means is the more antibiotic present, meaning the more antibiotics that you pump into the incorrect people, the more advantage the resistant bacteria will have. Because like I said, what you're doing is you're killing off all the normal flora, which means you're leaving space and resources and nutrients for the resistant problematic pathogens to now replicate and replicate and replicate and to really flourish. Okay? So what this means is you're creating evolutionary pressure because only the most fit resistant bacteria will survive the antibiotics that you're giving, and they'll then pass on the resistance. Whereas all the other ones that are not resistant, they die off, so they don't get to pass on that resistance to offspring. Now, I want you to put stars next to that explanation because this concept of how exactly antibiotic resistance is coming about is really important because so many people have misconceptions about what is actually causing the antibiotic resistance. For instance, many people mistakenly believe that when you give an antibiotic to someone, you're then mutating the bacteria so they're resistant and you're making them stronger. You are not doing that. Okay, the bacteria themselves are not changing. They already have the antibiotic resistance, and so when you give that person antibiotics, only those resistant ones will survive, replicate, and pass on the resistance to more and more offspring. Okay, so please keep in mind when you write down your explanation for the second question, that concept of increased exposure, make a note that the bacteria is not getting mutated, the bacteria is not getting changed by the antibiotic you are giving. They already have the genes for this resistance. Now, the last thing that you see on this slide is the question, what is the biggest contributing factor to antibiotic resistance? And this is critical for all of you going into the medical field. Okay, the answer to that question is in big, bold, red letters on the very next slide. So here it is, the biggest factor that's contributing to antibiotic resistance is the inappropriate clinical use of antibiotics. So you have to ask yourself, what exactly do we mean by this? What is the inappropriate clinical use of antibiotics? Well, that's a fancy way of saying misuse of antibiotics. And there are a lot of examples of this that I'm sure all of you are familiar with, either in your clinical experiences or your own personal experiences as people who have, again, at one point or another, dealt with antibiotics or taking medicine. The number one example of this is prescribing or taking antibiotics for a virus. Because what will the antibiotics do for the virus? Absolutely nothing. Instead, what will the antibiotics be doing in that person? Not harming or killing the virus. Instead, the antibiotics will be killing their normal flora. And as you kill the normal flora, what happens? The ones that are resistant will flourish, take over, replicate, and pass on the resistance to their offspring. The second big example of inappropriate clinical use of antibiotics is, how many people do you know, as soon as they start feeling even the slightest bit better, what do? They stop taking their antibiotics before finishing the full course. This is a big problem. You don't want to do that. You want to completely finish the antibiotics and make sure that the infection is fully killed off. Otherwise, you are just lowering the amount of normal flora while allowing the pathogens to kick back up into bigger numbers. Okay? And as you understand more and more about infection cycles, you'll see that yes, you will start feeling a little better while you still have plenty of that bacteria still in your body. Okay? So never stop taking antibiotics early. Finish the full course. And the third thing, never, ever, ever do is never use prescriptions that were prescribed for someone else or even for yourself, but back in the day, let's say for an old infection you had. Okay, because how many times do you see someone, let's say, will get some dental work done, they have some antibiotics, they didn't fully use them because they broke our inappropriate clinical use bullet point number 2 we were just talking about. And then what do? Year or two later, I'm not feeling so well. Oh, I think I might have an infection. Oh, let me grab those old antibiotics from my cabinet. What did they not do? They did not get cultured to figure out what exactly they have. Do they even have a bacterial infection that those antibiotics will do something for? Well, it's not a full course of antibiotics, so it's not going to do what it's supposed to anyway. And odds are, how many times is it just a viral infection? So since they didn't go to a doctor and get a new prescription, they're now taking old pills that probably won't even work for what they have. Okay, and again, what are those old pills doing? Killing off the normal flora and allowing the resistant pathogens to flourish, replicate. The last point that I want to make is that hospitals and healthcare facilities are ideal for the development and spread of antibiotic resistance. Because what do? They are constantly pumping antibiotics into all patients And many times they have not cultured to see if they're using the proper treatment for the proper infection. And again, how many of those patients actually just have viral issues? Okay, so when they're doing things that they consider preventative or protecting the patient, in the long run, sometimes they're actually hurting that patient. Okay, so yes, antibiotics are a very good thing when you have an infection that actually requires them. Okay, so please don't take this lesson and say, oh no, antibiotics are bad, I'm never going to take them, you shouldn't be giving them out either. No, antibiotics are very, very good if you have a bacterial infection for which you have been cultured for, and we know exactly what to target with which antibiotic. Okay, what we're saying is the misuse or improper use of antibiotics is what's the problem here. Okay, now to recap the different misuses that I've mentioned so that you can jot them down and further explain them in your notes. The first one we said, prescribing or taking for a virus. Okay, and make sure you understand why that's such a problem. What will that do? The second one is, like we said, people tend to stop taking antibiotics early as soon as they think that they're feeling a little bit better. Third one, people use what was prescribed for someone else or even for themselves, but back in the day, old prescriptions for themselves. And the last one that we mentioned is hospital and healthcare facilities overuse and don't always culture. So they're the perfect breeding ground for antibiotic resistance. Now we keep saying the term antibiotic resistance, and we haven't really gone into the details of what exactly that means. What mechanisms are these bacteria using in order to fight the antibiotics? Okay, there are 5 main ways that bacteria will resist antibiotics. The first one you see here is by blocking the drug's entry into the bacteria. Okay, so if the antibiotic cannot get into the bacteria, it can't kill it, right? The second one is inactivation of the antibiotic. So in this case, the antibiotic makes it into the bacterial cell, but the bacteria will then break or destroy the antibiotic. The third one is my personal favorite, and it's efflux pumping of the antibiotic. What this means is that as you pump the antibiotic into the bacteria, they then keep throwing it right back out of their cell, okay? Pumping it out so that they never have a toxic level of the antibiotic within them. The next one is the modification of the antibiotic target. And what that means is, in some bacteria, whatever structure would have been targeted by a particular antibiotic happens to be slightly a different shape. Okay, so now that antibiotic is unable to bind to that target in that particular bacteria. And if the antibiotic can't bind, it can't do its job at destroying the bacteria. So the bacteria is now safe. The last one is alteration of a metabolic pathway. And whenever we say metabolic pathway, which one do you want to write down? Folic acid pathway. Okay, the folic acid pathway is the big one. So in this case, if the bacteria can find an alternate route of getting folic acid or whatever the metabolic pathway is, they can survive. Okay? What I want you to do is next to the second bullet point here, inactivation of the drug, I want you to write penicillin, okay, which is the famous one that we know, as well as cephalosporins, which you'll see the spelling in a few slides. For efflux pumping, I want you to think of tetracycline. Okay? Now we're going to go over each of the mechanisms of resistance in more detail now, and that way you can really get a visualization of what's occurring. We're going to skip the blocking of the drug's entry and head right into inactivation of the antibiotic. But before we do that, I just want to once again point out that with any of these mechanisms, the goal of the bacteria is to survive the antibiotic, to not get killed off. So the first mechanism of resistance I want you to know is the inactivation of the antibiotic. So in this case, the antibiotic will enter the bacterial cell, but what the bacteria will do, it will have some sort of enzyme that can then break that antibiotic. Picture it cutting it up or cutting the main part of it, so that antibiotic cannot occur, cannot do its job. Okay? The big example of that is penicillin, okay, which you've seen in labs, in microbiology lab class. Okay? With that inactivation of that particular antibiotic, of penicillin and cephalosporins as well, you have the idea of the enzyme being beta-lactamase. And the beta-lactamase will cut the beta-lactam ring of penicillin, or the penicillin family of drugs. And if that is cut, then the penicillin can no longer bind to the bacterial enzymes that form peptidoglycan. And so their cell wall is safe. Okay? In addition to penicillin, this also works for ampicillin as well, which is the one that you've seen in lab classes. Okay? You'll see this in more detail in a few slides where you'll be able to see penicillin and cephalosporins specifically. The next example is the one that I said is my favorite, and for this one I want you to think of tetracycline. Efflux pumping simply means that the bacteria is actively pumping out the antibiotic. So every time that antibiotic enters the bacteria will have porin channels, okay, protein channels on their outer cell membrane or cell wall. And what they will do is every time the antibiotic enters the cell, they'll then push it through the porins to exit the cell. So they're always keeping the antibiotic levels in that cell at a very, very low, minimal level that is not toxic to them. The next example is modification of an antibiotic target. And for this one, this is the example I gave in terms of if the target in that cell happens to be a different shape, okay, or let's say missing a particular receptor, the antibiotic is unable to recognize and bind to it, which means the antibiotic can't do its job of killing the bacteria. That bacteria will survive. The big example of this is a lot of times you'll encounter bacterial cells with a slightly different shape to their ribosomes. So if the antibiotic is meant to bind to the ribosome, but that ribosome is the wrong shape now, the antibiotic can't do its job and that bacteria will survive. And notice how I say in the slide, you'll see the bullet point, mutate gene that codes for target protein. What this means is that bacteria will already have a mutated gene from its past through evolution, and that will code for the different target protein. Okay? Now, with that, it's not the drug itself doing these things. Okay? The drug is not changing the proteins or the ribosome in that cell. Keep in mind, it's just that the bacteria already happen to have a different shape, a mutated protein, and that was a benefit when the antibiotic then encountered that cell. The last mechanism for the resistance of antibiotics is alteration of a metabolic pathway. And like I said, for this one, always think of folic acid synthesis. Now, when we talk about folic acid, the first thing you should always ask yourself is what exactly is folic acid used for? Now remember, we said folic acid is used to make nucleic acids, such as DNA and RNA, and that is critical to bacterial and human cells. So think about it. If you have an antibiotic that is trying to kill a bacteria by destroying its ability to produce or make folic acid, then how can that bacteria overcome that and survive this attack? Well, what the bacteria can do is have an alternate pathway. If it has some other way to obtain folic acid, then it doesn't matter if that antibiotic tries to destroy its regular pathway. It still has folic acid, so it will survive. The example that I usually use for this is some bacteria happen to be what I call human-like. Some bacteria have the ability that we have, which is to take in folic acid from their environment, from the media they're in, and use that rather than having to make it. Okay? So you'll try and kill that bacteria by destroying its ability to produce folic acid, but the bacteria won't care because it has the ability to just grab more folic acid from around it, from its own environment, the way we do, like when we eat leafy greens or take prenatal vitamins. Now, the last thing I want to do over here is to give you some specific examples of antibiotic resistance. And for each of these examples, the first thing that you have to do is think about how that antibiotic would try to kill the bacteria, and then think of how that bacteria would fight back. Now, before we go into each of those details, I want to mention one more thing. The first specific example that you should know for antibiotic resistance is how bacteria can resist penicillin and cephalosporins. Now, like I said, the first thing you have to ask yourself is, how would that antibiotic normally kill a bacteria? So the way penicillin and cephalosporins work is, remember, they have that beta-lactam ring, which will then bind to the enzymes that would normally build peptidoglycan in bacteria. So they block the NAG-NAM formation, or they block peptidoglycan formation. So now that bacteria can't make its cell wall. Boom, that bacteria is dead. Okay, and again, this is a reminder of what that beta-lactam ring looks like. So how would a bacteria resist this antibiotic? Well, what if it has the plasmid, like I just said, that can code for beta-lactamase, meaning what if it has the gene that ultimately makes the enzyme that cuts this ring open. And when this ring is open, cut open, that can no longer bind to the bacteria's enzymes, and it can no longer kill the bacteria. Now keep in mind, which bacterial infection won't penicillin work for, and won't this resistance matter about? Well, remember we said mycoplasma, okay? Mycoplasma, or walking pneumonia, none of this slide matters for, okay? And keep in mind that I always say which bacterial infection won't it work for to target the cell wall. So never say, oh yeah, penicillin won't work for a cold. No, that's a viral infection, okay? So I'm asking for which bacterial infection won't penicillin and cephalosporins work? And the answer is mycoplasma, walking pneumonia. Okay, and we already explained that bullet point of how exactly beta-lactamase is working. Keep in mind, it's inactivating, it is cutting that antibiotic. The next resistance that you have to know is how bacteria resist chloramphenicol. But like I said, the first thing you always want to ask yourself is, how would this antibiotic try to kill the bacteria? And the way chloramphenicol works is it's an antibiotic that inhibits protein synthesis. So what it does is it will bind to that rRNA of the ribosome and block the reaction of building the protein. Now the way it does that is through this OH right here. So this whole structure is chloramphenicol, and it has this OH here that allows it to bind to the ribosome and block the bacteria's protein synthesis. So how would a bacteria fight back? Well, change this block this OH so that it cannot bind to the ribosome. Okay, so what some bacteria are lucky enough to have is an enzyme that allows it to inactivate the addition of acetyl groups to that OH. Okay, fancy way of saying that some bacteria will have an enzyme that makes the OH into this structure, okay, or even changes this OH to this structure, so that now these versions of chloramphenicol do not have an OH to bind to the ribosome. Okay, so if I ever asked you about how a bacteria would be resistant to chloramphenicol, that bacteria can have an enzyme that modifies the OH, blocks the OH with an acetyl group. Okay, so it's capping that OH with an acetyl group so that antibiotic can no longer bind to the ribosome. Okay, the specific name of that enzyme is chloramphenicol acetyltransferase, or CAT for short. You don't have to memorize that name. Just know that to resist chloramphenicol, a bacteria can have an enzyme to cap that OH with an acetyl group. The next category of resistance is very, very similar to chloramphenicol, and this is the resistance to aminoglycosides. Now again, the first thing you have to ask yourself, how would this antibiotic kill bacteria? Well, this antibiotic, which includes streptomycin, kanamycin, neomycin, and some of the related antibiotics, what they do is they also inhibit bacterial protein synthesis. So they use certain groups such as OH groups or NH2 groups to bind to the bacterial ribosome, and that will block protein synthesis and kill the bacteria. So how can that bacteria fight back? Like we mentioned in the previous slides, by capping or modifying those groups that the antibiotic would use to bind to the ribosome. So if the bacteria can now cap the OH groups or NH2 groups with acetyl groups or phosphates, or somehow modify these groups, these OH and NH2 groups, that antibiotic can no longer bind to the bacteria's ribosome. It can no longer block protein synthesis, so the bacteria survives. Okay, so whenever you hear aminoglycosides, think resistance by modifying those binding groups. Okay, modifying the binding groups. Okay, and this can include, again, addition of phosphate, AMP, or acetyl groups, so that the antibiotic can no longer attach to the bacteria's ribosome. The last resistance to mention is the one that I said was my favorite, and that is tetracycline resistance. Now again, the first thing you have to ask yourself, how does this antibiotic work to kill bacteria? Well, what tetracycline does is it's yet another one that inhibits protein synthesis. Now, what the bacteria in this case does to protect itself against the tetracycline is it has a transport pump. So remember, this is efflux pumping. So as the tetracycline is brought into the cell then keeps pumping it out. And it's able to do that because it contains R plasmids, meaning resistance plasmids, with genes that have produced special pumps or channels on their outer membranes. And so they can actively pump out the tetracycline so that that antibiotic never reaches a toxic level. Okay, it stays low in the bacteria and they don't get killed by it. Now the last thing I want to cover is the take-home message, okay? And to once again remind you that the overuse and misuse of broad-spectrum antibiotics is a huge problem, okay? And to draw out why this is again, okay, to really reinforce what I've been saying this whole time, I've made a diagram on the next slide. Make sure to pause at some point and draw this diagram in your notes so that you have the take-home message of antibiotic resistance, which is that in your body normally, you'll have a whole bunch of normal flora, which we represent here in blue, and scattered throughout there'll be some resistant potential pathogens, but right now they can't do much because the normal flora is all over the place, taking up space, taking up resources. Now, what happens when you then pump antibiotics into that patient? Well, the normal flora that was not resistant to that antibiotic have now been killed off, and only the resistant potential pathogens survive. Now, what do you notice they can do? Boom. They now have all the space and resources to replicate. And now they flourish and they pass on to each of these offspring that resistance, right? Because that's how bacteria replicate. They're dividing themselves. So the offspring will be identical to them. Okay, so now they have the chance to reproduce and pass on that resistance. Okay, so again, keep in mind when you hear overuse of antibiotics or misuse, think of the fact that it's killing off normal flora, so the resistant pathogens now have a chance to flourish and pass on their resistance, okay? Notice there was no mutations occurring here, no, strengthening of the bacteria themselves. They were already strong. They were already resistant. We just gave them the ideal environment to pass on that resistance and to flourish. And that is it for our semester. It has been an absolute pleasure to teach, well, most of you this semester. Just kidding. All of you. I really enjoyed this class, and now for the rest of your lives, any questions you ever have, you are always more than welcome to contact me. Now, I know you probably can't hear me over your tears and sobbing, so sad that this course is ending, but I do wish you all the best of luck throughout the last few weeks of the semester, as well as your future careers and all future endeavors. And one more time, I get to say, it is now time to head over to that discussion board, post your questions, help yourself study and review, okay? And as always, any questions, any concerns you have, just send me a message and remind. I'm always happy to help. Okay? Have a great day.